recent research works to the colleagues and friends. And thanks, Remy, the introduction, and many thanks for the WHEAT initiative for the invitation. And today my topic is WHEAT disease resistant genes from NRR to WTK. So next, please.
Okay.
So everybody, we are here that we are working on crops that we know the impact of the crop disease to the food society. This is one of them. My favorite slide. I want like to every time that I show to my students, I would like to talk to them that the disease resistance is impact the human civilization and society. That is the famous one caused by the potato blast disease that caused the millions of people died and migration. So next, please. So wheat is one of the most important food crops in worldwide, and also is very important in China. Next, please. So next one, please. So for winter wheat, that we have most of this longest time for in the growing time in the field. That's like we have it in autumn. We sow in the seed in autumn season and passing the long winter, and then in the springtime and harvest in summer. That means in the fall seasons for wheat, we can see them in the field. And that means also the wheat plant can be suffers the most kind of disease traits that cause a lot of loss in yield and also in grain quality, such like the pod mildew. There are three kind of rusts and the spot blast and also the fusarium. So next, please. So in China, that we can see here in the past 20 years, strap rust, the leaf rust, the pod mildew, and the fusarium head blight, their impact and the and the damaged area and for each year. We can see here that the strap rust is used to be the most serious of the disease in China. But thanks for the breeders that we developed a lot of resistance cultivars, and so this strap rust is basically under control. And the fusarium head blight is getting even more kind of serious recent years. And pod mildew is the top in annual damaged in every year. It's around kind of 6 to 8 million hectares for each year. That is the top impact area in general. So next, please. So especially that when this for this kind of pathogens for wheat, especially for the biotrophic fungi like rustus and pod mildew, it's kind of the virulence isolate. It's occurred very frequently, so it can be overcome the disease resistance gene in the host cultivars. For example, this the new virulence isolate for the strep rust, the V26 or CYR34, it overcomes several most important strep rust resistant gene like YR10, 24, 26, and another one that is in the cultivar TRAMA42. So that's it's caused we lost a lot of the resistance in many of these kind of commercial cultivars. So seeking new resistant genes for breeders is a very urgent task for all of the wheat researchers. So next one, please. So normally everybody we hear we know for wheat breeders and scientists that we can have three kind of layers for disease-resistant genes resources. We know that from the commercial cultivars, it's very narrow genetic diversity for the disease-resistant genes. So we are going back to the landrace and the wild progenitors and the relatives to find more diversified disease-resistant genes and transfer them to wheat and for breeding newer cultivars or germplasm and use them for commercial breeding. So next one, please. So in past several years, we are working mostly on powder mildew disease resistance and rust resistance. And we found several of these genes, like powder mildew resistant gene 5E, 26, 41, and another genes like it's PM60 allele, and the leaf rust resistant gene LR13, and also the same as the hybrid necrosis gene NE2, and also the barley strep mosaic virus resistant gene called BSR1. All of these genes are NLR kind of resistant protein, and also that's the major player for disease resistance. Like we know that it's also found in rice and also in Arabidopsis and also other crops. It is a major player for disease resistance. So we have want to show our works for of this kind of a story we worked and to see how they are looking like.
Oh.
Okay, please. Yeah, next one, please. So the PM5E is was found in a Chinese wheat landrace, and it was originally being mapped on the chromosome 7B in the long arm, and it's come from a landrace and also integrated into breeding germplasm, like in Fuzhou 30 and in Tangmai 4, they are be used for breeding. But after we have the gene, we found that there's in commercial cultivars, there's no gene of this PM5E was found in the commercial cultivar. So please, next one. To map this gene and for cloning it, we started in more than 10 or 15 years ago. When we just back to China, we are working on that. Back to 2000 and almost 15 years ago. So on that time, we have no reference genome sequence of wheat. So we use every kind of method to develop markers to find mapping the gene. So we use a two-step block segment analysis plus RNA-seq approach to develop markers linked to this gene. Next one, please. And finally, we map this gene in a very small genomic region. It's about 13 kilobase pair, containing 3 of these genes. For the 3 of these genes, that in the first 2 is what we call it by NLR kind of disease resistance, and the last one is called C2 dominant protein. And so between the resistance and the susceptible parental lines, we can find no sequence variation for the C2 and also no sequence variation in the Chinese spring. So we think this gene possibly not involved in this polymyl resistance. Then we try to see how about the first 2 genes. So please. Then we did transformation for these first two genes. You can see that, okay, next one, please. You can see that when we transfer the first gene, it's called RXL, into a susceptible cultivar filter, you can see is a full susceptible. In other way, that when we transform the second gene, this NLR gene, into the susceptible cultivar filter, that we can see either in its native promoter or in a ubiquitin promoter that give full resistance for pod mildew. That means the second gene in the middle, that's NLR gene, is the PM5E. It's encoded a NLR receptor. Next, please. Then we want to see, since this PM5 locus, they have already several of these allelic variations like 5A, 5B, and 5D, and the others. We want to see how about this allelic variation. Then we tested more than thousands of lines or cultivars from worldwide collected, and we found this there are several different kind of sequence variation between these different alleles. And very interestingly, we found this PM5E, there has only one single nucleotide difference from a susceptible landrace we call BM. There's only a single nucleotide difference. And compared to other like Chinese spring and PM5B and other kind of different assessments, there are some kind of more sequence variations. So next one, please. So we have a look for this full gene sequence in this area for genes, RXL, and this NLR and the C2, this 13 kilobase pair in different kind of assessments. We want to see the detailed sequence difference. So very interestingly, we find that there is the single nucleotide variance in this PM5E gene. There's a single A to G variation in this position 3033. In this reason, if there's a nucleotide that you give a full resistance for the pod mildew. If this other one, that is G and the other variations, is given a full susceptible. So based on this variation, we design a custom markers. So next, please. So for this custom marker for PM5E, we can fully screen the wheat germplasm, and it can be used very quickly. You can find this gene. It can be used very reliably and efficiently in a marker assistant selection in the breeding program. So next, please. Okay, that's the story of this PM5E. And another one is this PM26 or MLW 160. This comes from wild emerald wheat. This PM26 gene is discovered by Dr. Junkang Rung, and when he was did his research work in Israel in Dr. Feldman's lab, and he found this gene, the PM26, is on the short chromosome of 2BS. And also, our lab that we found another assessments of wild emerald wheat. We found this, some of these polymer resident genes in the same kind of genetic region. That's like this MLW 170 and also this MLWE74. So they are all mapped in approximately the same kind of region. And then we did a fine genetic mapping for these genes, see if they are in the same locus or a different allele or a different locus. Please, next one. So after we develop a very fine genetic map, we found this is PM26, this MLW170, and even also this MLW74. They are all located in the same kind of genomic regions. So in the same kind of marker, we can anchor them in the same area. And so we compare it in other available genomic sequence, like in wild emerald in Dravidian, we find this area there are more than a couple of these kind of NRR protein was annotated in this genomic region. So next one, please. Then we compared other genome sequence available currently, like Chinese Spring and the 10 plus genome, and all other with genomes or with relatives. You can see in this region, it's kind of very rich in this resident gene area. So we amplified all these sequence and all their homologs in our resistant assassins. And unfortunately, we didn't got any kind of useful information from all these available genomic sequence here. So next one, please. So then we think about other method. See if there is kind of genomic structure variation in this area. Then we applied. Then we yes. Okay. Then we applied the PacBio sequencing, and to see if we can alignment or assemble this genomic area to see their difference or not. Then we applied the PacBio sequence, and we sequenced the 4 cells, and then we assembled these this tetraploid genome, and we got several of these contigs. And we got this from the anchored marker. We have these contig that cover this entire region. So next one, please. So after we have the full annotation of these contigs in this minimal genomic region for this gene mapping, and we found there's 2 more of these NRR genes compared to all other available genome sequences. So next one, please. And then we try to see if these 2 of these genes, or which one is the PM26. Since we have developed a lot of these EMS-induced mutants for this PM26 and also these wild emerald assassin IW170, and we can see if all these additional NRR genes, CNL1 and CNL2, if they have sequence variation in the mutant. Then we find this CNL1 have a lot of mutants enriched in this gene, in these 2 of these 2 assassins mutants. That means this CNL1 is the PM26, and so from the sequence available that we cannot get it, so we can using the currently available is PacBio long read sequencing to have this additional one. So this means that have a present absence variation for this PM26 genes between this wild emerald and also other deployed on the hug side. Okay, next one, please. Next one. Okay, so the next story that is PM41 is also from wild emerald wheat. For this gene that we mapped this in this chromosome 3B back to 2014. It's in a very small genomic region, and on that time we think that the Chinese Spring chromosome 3B genome sequence is ready, So we are very happy for that. We think that we can get this gene. But when we look at the fun dynamic region in these 7 genes, none of these genes was looked like the resistant gene. So that and then we on that time we still have no other sequence available. We develop a back library for these wild MR assassins and try to develop a physical map to clone it. Okay, next one, please. Next one, please. Then we sequence the bikes from the back library and perform a chromosome walking and to have the full sequence in this area. And after we sequence the bike and the sequence assemble the bike sequence, we found there's one more NLR genes compared to Chinese spring. There's sequence deletion, and after that we the wild MR with J-Witten sequence is available, and then we compare that. We found there's a non-functional allele of this gene in the NLR protein. And then we look at how about these mutations of this NRR for the susceptible stability. So we can see for this susceptible mutants of this NRR that you can see the loss of this resistance. Okay, next please. And we then perform a transformation of this either ubiquitin driven by ubiquitin promoter, and also next one, and also driven by its native promoter. We can see you have the full resistance. Next one for this NR protein. So that means this NR is the PM41. So next one, please. Then we want to have a look for this where is this gene distributed, and we found it's only available or present in this fertile crescent area in the southern wild MR population, only in the northern part of the Israel or around that region. It's co-located with another 2 genes, that's WIR15 and WIR36. They are almost co-located in the same area. So next one, please. Then we have a observation of these NR genes in the natural populations, like in wild MR wheat, tetraploid, cultured wild MR, and drum wheat, and also common wheat. We to see how about this allelic variation or haplotype variation of this gene. We can find like in Chinese spring, there's a null allele. That means a sequence deletion of this resistant allele. And but we found some other assassins, like in the drum wheat in London, that's kind of a HAP2. This haplotype, they have a full length of this coding region sequence. And other kind of haplotypes you can find in other kind of in tetraploid and hexaploid or common wheat. So next one, please. But from the wild MR wheat population, we can find all of these 7 haplotypes in the wild MR wheat populations. But when you test it in the drum wheat and in the hexaploid or common wheat, there's only 3 kind of haplotypes. That means the polyploidization and domestications bottleneck that is also for this gene or for this locus. But very interestingly, we can find there's more than 90%, the red one, this HAP2, of this drum wheat and hexaploid or common wheat. They have more than 90% of these wheat assassins containing this HAP2. This HAP2, they have a full line coding sequence, but it's susceptible. The next one, please. So we are wondering why this difference. Then we have a look for this HAP2, and we call it a PM41B allele. So we can find this allele is they have a full line coding region, but it's not expressed. Next one, please. Because of these two transpositions in the promoter region and in the 3' side. So these two transposition insertions are silenced of this PM41 allele. It's not expressed, so the loss of resistance. So next one, please. So then we are wondering if we can express this gene sequence, can restore the powder mildew resistance. Then we transform these two, this coding region, the genomic region, and also the CDS into susceptible cultivar filter. We can see there is a powder mildew resistance for this silenced PM41B allele can be used for wheat disease resistance. When we look at it in the full genome of the wheat, we can find a lot of these kind of genes. They are silenced by the transposition in the genome, but they are still there and sleeping. So next one, please. We have a very good symbol for this one. It's like a sleeping beauty in the we can see in the book or in the film, like this kind of gene. If you wake up this gene, you can have a resistance or restore the resistance using a commercial cultivar, since this haplotype is very common in the wheat and the tetraploid and hexaploid cultivars. The next one, please. Next one.
Sorry. I think I pressed
Okay.
admit
Yeah,
all.
okay. Yeah, that's another one. That's also there are two of these powder mildew resistant genes that we find in wild emerald wheat. We call them in two of these allele that's mapped in the same area of this chromosome region, and it's in the 7A region. We know that in this area there's several of these powder mildew resistant genes already mapped here, like PM1 and or other alleles were temporarily named genes in there, but we don't know which gene is the same of this, is different or the same allele with others. So we have this temporary naming for this one. Okay, next one, please. And it's very hard for us to isolate these two genes. So we applied another one approach. Please, next one. And we use a bulk surrogate association with another sequencing approach, like a ChIP-seq, in collaboration with Dr. Yijing Zhang from Fudan University now. And we try to enrich this kind of NRR fragment between the resistant and susceptible blocks. And very luckily, we have a lot of this kind of NRR fragment was enriched in this. We can see from here that in the chromosome 7A region, and several of them, and we can see is all of most of them is located in this reference genome region in this mapping area. And next one. So we have one of these contact sequence. It's very similar, very highly similar to the PM60 gene that's already cloned from Warao to wheat. So we guess this gene possibly is also linking to our genes here. So next one, please. Then we use this kind of NRR fragment to develop several of these markers. And we found most of these CGT kind of markers developed from this NRR fragment was co-segregated or very closely or tightly linked to the two genes like ML WE18. From this method, we can map these two genes in the same kind of area. And also, we can see from this PM60 homolog context, we can have a look if they have a sequence there, and we can see in several of these EMS-induced mutants. Next one, please. We can find they have in this PM60 homolog sequence, they have a lot of mutations. And also, next one, we developed the transgenic lines using the native promoter of these two genes. We can see they get full polymerase resistance from these two alleles. So that means these two alleles from the wild amyloid is the new allele for the PM60 polymodal resistant gene, which come from triticum without. This is a new allele that's variations from the tetraploid wild amyloid. So next one, please. Next one. Okay.
Oh, is it we are?
Okay. Yeah. Okay. Then we are working on another one. That's a leaf rust resistant gene. It's ERA13 or ERA-GH22. It comes from one of the very famous Chinese commercial wheat, like Zhongmai 22. It's highly resistant to strep rust and also to leaf rust. This work was collaborated with Dr. Zhaifeng Li from Hebei Agricultural University. And he was focused on the leaf rust resistance side. And at the same time, in our lab, we are working on this necrosis gene. We call it ELS1 or E2. So next one, please. Next one. Okay. For this early leaf synthesis gene, ELS1, we call that on that time, we found it's have a very strong phenotype and similar to this hybrid necrosis. And it's very, after we map that, the next one, we found this gene is in this same gene, same region, like in ERA13 and E2 genomic region, which worked by Dr. Peng Zhang and Bob McIntosh in this chromosome 2BS. They are also working on this gene. And luckily, we have one of these markers we call WGGB302. It's co-segregated with this early leaf synthesis. And so then Bob and Peng they tested this marker, and they are graduate student team, and tested this marker. They found that it's also co-segregated with this ERA13 and E2 gene. So next one, please. Then we found that we can develop a map-based cloning of this ERA-DH22. There's only one single MBS LRR gene. So next one, and from the transgenic complementary and also from the EMS-induced mutant, also we hybridize this NRR transgenic line with our leaf synthesis lines. We can confirm this is a hybrid caused hybrid necrosis. So after we got this NRR receptor, that we can know that this gene, this ERA gene from the Zhongmai 22 cultivar is the same as ERA13, and is the same one as this very famous hybrid necrosis gene NE2. So next one, please. Okay, the last one for this NR protein is we are working with a former colleague from China Agricultural University, Dr. David Li, and also Andrew Jackson from UC Berkeley. And let's back to more than 10 years ago. At that time, the brachypodium was proposed as a new model system for the monocot plant, like especially for the grass family. Since this barley streptomotic virus is very commonly used in many kind of life as a virus-induced silencing system, we want to see if there are kind of a resistance cultivar or lines were resistant to these virus. But interestingly, we found the two of these lines, like the BD31, the Brachypodium line, is resistant to these virus, and the sequenced one, the BD21, is susceptible to that. Then we developed a fine mapping population, and we mapped this gene in NRR-rich region also, and we got the candidate one. We did transformation in the help of Dr. Jia Wu and Dr. Lin Fu to the susceptible Brachypodium lines. We can find the overexpression of the gene can restore the resistance to the barley streptomotic virus. And also, we transfer this gene into wheat and barley also, and we can see this gene also works in barley and also in wheat, resisting to virus. For this gene, we did a lot of other works, and it can cause leaf necrosis in the tobacco leaves. And the next one, please. And also, since Dr. Dawei Li in China Agricultural University, they are working on the BSMV side. They clone the effector TGB1 gene, and we found this BSL1 protein can recognize this BSMV effector TGB1 and cause a gene-for-gene interaction and between of this disease-resistant gene and the BSMV effector one. So this is the first time we can isolate a NR protein in monocotyl plant can confer resistance to virus in monocotyl plant. Since before that, we know that in like in tobacco and in orbitovices, there's a lot of other virus resistant genes are already isolated. They have this kind of NR protein, but in monocotyl plant, like in rice, there's never we've never found an NR protein was resistant to a virus. And this is from this gene, this BSL1 from Brachypodium. This is the first one. This NR protein is from a monoclonal plant. Okay, next one, please. Okay, that's the six stories that our lives we worked before this for this NR protein and the major player for the disease resistance in wheat. And the second big group is the tandem kinase protein. Recently, for our lab, we found there are new players also for the disease resistant gene in the treated crops. Since this kind of protein, they have two kinase fusion together at the same gene. And the wheat one, we found that we call them wheat tandem kinase in sort of WTK. The first one is the strep gene YR15, called WTK1, and the second one is the stem rust resistant gene SR60 WTK2. And the TM24 gene, we call it the WTK3. And recently, there's a new allele of this gene, the DSC lab from Paul's lab. They found it's also resistant. It's to the wheat blast. And also they have the new one, WTK4 is from Agilops torsii. It's also resistant to powder mildew. And very recently we found the PM36 from Werner Emmer wheat is also a WTK. And the leaf rust resistance LR9 and also LR58, because they found is also is a WTK. That is recently reviewed in Nature genetics papers. Okay, next one, please. So we will have a full look of these two genes. Okay, this PM24 is also from a Chinese wheat landrace. We inoculate many kind of powder mildew isolate on these three assassins of the like Xiaochao, Baihulu, and the Hulutou. And we found they are all resistant to the avoidable or inoculated BGT isolate. And the cross between these three assassins confirmed that they are located in the same locus. So next one, please. So after perform fine genetic mapping, and we found this PM24 was located in a genomic region containing 6 of these putative genes, and 2 of these them are causing our attention. That's one of these is a kinase protein, and another one is a protein, and we frequent variation the resistant lines and also the susceptible line between these 2 genes in the resistant and susceptible parent line. So then we have a look for which one is this of genes is the causal gene for this PM24. Okay, next one please. Then we perform a transgenic validation. We can see from this either under native promoter for this WTK and the NR, you can see only this WTK gene. You can have the resistance phenotype, and the NR genomic DNA transformation in filter, you cannot get a resistant phenotype, and also the overexpression of the WTK3 CDS sequence in filter, you can also have a very strong resistance to portal mildew. That tells us this which tandem kinase protein 3 is the causal gene for this PM24. Okay, next one, please. And in the same time, we can see from this EMS mutant, we can have a lot of these mutations in this with tandem kinase protein. Is in underside confirming this one is the PM24. So next one. Then we have a look for this allelic variation of this gene. We can find this a lot of kind of sequence variation for this allele. It's very diversified variation in hexaploid, tetraploid, and also in Adelopes Taussii family. So there's a lot of sequence variation. They have very rich genetic variation for this locus. So next one, please. Then we have a look for the full line sequence of this gene to see where is the key sequence can cause the resistant and susceptible. And from all these sequences, we can find there is in the resistant plant, and also in 6 in 4 of these things, we can find the 6 nucleotide deletion in the resistant plant, and all the other available either in diploid Taussii Adelopes and also in this hexaploid common wheat. They have all of these 4 nucleotides there. So this is a very key sequence variation between the resistant one and the susceptible one. And we also developed some custom markers for this one and to use them in the marker system selection. So next one, please. Okay, that's the first one. We found that with tandem kinase gene can confer portal mildew resistance. And another one is the PM36 gene. It's also from a wild emerald wheat. And we can transfer them, after we transfer this gene into hexaploid wheat, we can see after inoculated with 104 BGT isolate, they perform all of them can resistant to all of these tested isolate. It's a very broad spectrum resistant gene. Okay, next one, please. Okay, so next. So, so we spend a lot of time to for cloning this gene. Since you can see from back to 2018 that we have already have only very small region containing only 3 of these genes. For the, this gene in the reference sequence, like in Chinese spring. Okay, and also after the other sequence available, we can see compared all others, there's no other genes available in this genomic area. Then we can after we doing RNA-seq, we can see there are only 2 of them were expressed, and we did a transformation. Okay, next one. And so both of these genes is expressed gene after the transformed into a susceptible filter, which cannot give us a full resistance. They are all still susceptible. So this, the next one, please. We are wondering where is the gene? We are wondering if there are still genomic or structural variations in this area. And then we recently performed high-fi seq and we sequenced 5 cells for this tetraploid which we generate very long contigs in long read, and to assemble after some of that we got contig can cover this region. So next one, please. Okay, next. Okay, after we okay, next from one.
Oh, sorry. Yeah.
Okay, after we have this contig, we annotated genes in there. We can there's more than 900 kilobits per insertion we find in this resistant assessment of the wild emerald. So that means there's a very large sequence variation in this area. After we from the RNA-seq result, we found there's only one of these additional genes beside of this former 2 of these expressed gene. There's one of these wheat tandem kinase gene is expressed. Okay, next one. Then we use virus-induced gene silencing with also we tested the genomic sequence variation of this EMS-induced mutant for this gene. We found that this WTK have a lot of induced mutations in this gene body. So that means this gene likely is the PM36. So next one, please. Then we perform the transgenic analysis, and we got the transgenic line recently, and we can see this after this WTK transformed in filter is get very strong resistance to powder mildew. Okay, next one, please. And this gene containing 2 of these fusion kinase domains there, and both of these 2 kinase domain is necessary for this its function, and also it has a is associated to one kind of the memory in the cell. Okay, next one, please. And also we developed a functional marker to test if this where is located. We can only find this gene in the wild emerald population in like in Israel or nearby regions. Okay, next. Okay, so in summary, that from our recent work, so we can see gene from like PM5E, 41, R13, and the BSR1, and also PM26. They are all belongs to the risk-specific NR protein family. Next one. And for this WTK, we clone the PM24 and the PM36. They are all belongs to tandem kinase family, and they exhibit a very broad kind of spectrum to resistant to all kind of BGT isolate we tested. Okay, so next one. And also we transform this kind of disease resistant gene like PM5E, 24, and 36 to the currently very elite commercial wheat cultivar to develop the new germplasm or new cultivar or new lines, breeding lines for using marker resistant selection, and also in combination with traditional breeding approach. This kind of line is ready to be for the breeders, can be used in their breeding program. Okay, so next one should be this should be the last slide. Okay, that's acknowledgement. So a lot of this kind of work was helped by many of the colleagues in China and also in overseas scientists from in the United States, in Australia, and also in CIMMYT, other places. So many thanks for them helps. Okay, next one. And also, I'd like to thank to my former students and former lab members and their current students working in the lab. And without their hard work, we cannot get this already result. So thanks for that.